Bon, et je pense que je vais commencer. Ok, bon, bonjour à tous et merci beaucoup pour être là. Et je suis ravi de vous présenter Shayla Salzman. Shayla est un postdoc à l'Université de Cornell, New York. Et elle a une carrière spectaculaire. Elle a fait beaucoup de choses. Donc, elle travaille avec des cycades ou cycadées. Ici, il y a une plante. Voilà, c'est de belles plantes. Il y a la maison. Et aussi, donc, elle a une carrière exceptionnelle parce qu'elle a fait beaucoup de recherches sur la biologie des plantes très intéressantes. Elle a fait son bac à l'Université de Californie à Berkeley. Après ça, le doctorat à l'Université de Harvard, Massachusetts. Donc, dans sa carrière, elle a publié beaucoup d'articles, plus de 15 articles, beaucoup de financements et beaucoup de présentations. Elle fait beaucoup de vulgarisation de la recherche aussi. Et aujourd'hui, elle va nous parler de sa recherche. Ici, quelques articles. Je pense qu'elle va nous parler de tout ça. Donc, on est très content de la voir. Et merci beaucoup, Shayla. Thank you very much, Shayla, for being with us. And if you don't understand, no worries. Probably nobody did. But anyway, thank you.
Yeah, let me just continue. Let's see. All right, there we go. And everyone can see that? You've got it all up? Okay. Great, thanks for having me. I'm super excited. I wish that coronavirus wasn't happening for a million reasons, but one of which it would be great to be up there and see the campus and get to see everybody in person. You just saw this picture, and it's because I use this picture in every talk that I give, because I think it shows you a few things that are important about my study system before I even get started, and a few things that are important about me and my approach to science. So you can see here, this photograph was taken in the field by my husband. And you can see first off that the plants that I study are cycads, they're gymnosperms. So this is the reproductive structure. I'm looking here at an ovulate cone, which produces the eggs. The pollen cones, which produce the sperm, are housed on a separate plant. And I study pollination in these plants, which we don't often think of gymnosperms when we think of pollination. We usually think of flowers and visual signaling. But I also love this picture because my husband took it. And it means a lot to me, him being in the field with me. And also you can see my wedding ring here that was then later stolen when I was mugged in Brazil during some other field work, actually right before I met Juan Carlos the first time. But my approach to science is I really want to understand the relationships and the backstory to everything that you see in nature, and what phenotypes and what traits may be present or may be absent, and their history in them, that we see in the biodiversity that we study. And I think that relationships are inherently interesting to all of us, and I always say, as evidenced by the wild success of every romantic comedy ever made, no matter how ridiculous the plot or annoying the characters are. And I think that's because we have a lot of just innate questions we want to ask about species interactions and about relationships between people. How does the relationship work? Can we help each other? How did we get here, and where are we going? We want to know if someone has to change. This is my desperate attempt to be a little more relevant with some more recent images of people that may connect with some other viewers. But in fact, all organisms engage in a range of diverse interactions with other species, ranging from, when you think of symbiosis, you might think of the sort of a spectrum from parasitism to mutualism. And some of these things are more obvious. We have predation or herbivory and pollination services. But when you have long-term species interactions that exist and persist over evolutionary time, you can get some really great traits that emerge from this. So in this top right, we have here Bacella drepanalobium, or Acacia drepanalobium, it's been historically named. And this is the African ant plant. So this plant in Africa grows these domatia that the ants live inside, and the entire colony of ants is dispersed across the tree. And in fact, these ants protect this plant from large herbivores such as elephants and giraffes. We have obvious things like this. We have the cyanobacteria and algae together that can form a mutualism with fungus to create lichen. So essentially to create what we would consider a whole different organism. We have things, I have this as cyanobacteria. I put this up for Juan Carlos because there's a bunch of great stories with cyanobacteria and cycads, but I really put it up there as an image of bacteria. We know bacteria is ubiquitous in our environment, and it has lots of effects on ecology and even behavior of organisms. This last one is a really great example. This is the Fingaris butterflies. And what they do is a small instar, as an early instar, these caterpillar fall to the ground and they have to be found by ants. And the ants bring them into their nest where the caterpillars feed on the ants and are fed by the ants in a process called trophallaxis. So this species interaction has evolved so long over time that the life cycle of both organisms is completely dependent on the other in this really bizarre behavior. So if we go back to those same questions that we can ask about relationships, we can ask those same things about species interactions in nature, but now we might use slightly more science sounding words. So we might ask, what are the mechanisms that actually keep us together? What type of symbiosis are we? Where do we fall on that spectrum from parasitism to mutualism? What is our evolutionary trajectory? And has there been any evolutionary innovation? And I believe that cycads are the perfect system to study this in. I'm in good company. Knut Norstag is a very famous cycad biologist. Here's a quote of his that I love. I consider cycads to be the Rosetta Stone for plant biology due to the wealth of information stored within them. And I would add that they can be the Rosetta Stone for species interactions biology. And that's because they have a lot of great characteristics that can be used to that can be important in the study of this. They are an extremely ancient lineage with fossil records going back over 350 million years. In fact, they are the basal lineage of gymnosperms. They are a gymnosperm, which means that if you're studying pollination, you don't have as much of the floral visual signaling is not as important in this system. So you can really sort of narrow and focus your communication mechanisms. They are highly toxic, which means that any species that interacts with them has to deal with these toxins in some way. They have multiple highly specialized species interactions from parasites, herbivores, the cyanobacteria that Juan Carlos, I'm sure, has talked about before. And they have morphological adaptations to symbiosis, like the colloid roots with the cyanobacteria. And they are, what I study most is that they are brood site pollination mutualism, and I'll talk more about that in a minute. But I also love cycads because they're just incredibly beautiful. So these are photographs that my husband took at Montgomery Botanical Center. And it's a good time to mention that cycads are the most threatened and endangered plant lineage in the world. So lots of my work is done at Montgomery Botanical Center, which is a cycad conservation garden in Florida. They are also the most beautiful plants in the ecosystem, in my personal opinion. And so this is actually a hike with Juan Carlos years ago, but I just think you cannot be in a forest with these and not fall absolutely in love with them. But as I mentioned, cycads have highly specialized relationships with both pollinators and herbivores. And that allows me to ask really specialized questions about the behavior of these two organisms, and to understand that both of them exist on the spectrum of parasitism to mutualism. So I study specifically the weevils that pollinate in Zamia. I'm going to talk mostly about that. But I do a lot of comparative studies with the herbivores of Zamia. These are Eumaeus butterfly larvae that do great damage to the plant. But most of the work that I do with Eumaeus is done with a collaborator, Melissa Whitaker at ETH Zurich. And if you're interested more in those, she's a good person to reach out to. So I study the pollination. And we know that specialized coleopter pollination is widespread in cycads. So for many, many years, cycads were considered wind pollinated because the grandfather of cycad biology, his name is Charles Chamberlain, had said, look, guys, I know you see beetles and weevils on these cones, but trust me, they're wind pollinated, they're gymnosperms. So if you go back through herbaria records, you'll find notes that say, found weevils on cone, and the weevils and the beetles have been ignored through time. But we now know that it seems as though all cycads are obligately associated with pollinators. There may be some wind pollination in cycas here, but in almost all cases, they are obligately pollinated by insects. And in almost all cases, it is beetles, coleoptera, beetles or weevils. We do have some thrips pollinators in macrozamia, and there has been one moth pollinator identified in Cycas anatrotentis. And interestingly, I found a related species of anatrotentis on zamia in Florida, but I do not know if it's pollinating, although it did have pollen on its wings and legs. But we know that the specialized coleopter pollination has existed before the rise of flowering plants. This is quite an ancient relationship between cycads and beetles. So I have here the rise of flowering plants really, and some fossil evidence that's come out fairly recently of pollination here. And this is reviewed more in a paper I'll talk about a little bit later if you want more information on it. But I study the pollination in zamia for a number of reasons. Zamia is the neotropical lineage. So it is a lineage of cycads that's found with one species in Florida. It goes through the Caribbean, Mexico, Central and South America, down to, reaches as far south as Bolivia. But also because of some really incredible work that was done in the 80s by two different groups of researchers. So Willie Tang and Knut Norstog and pals were both working on two different species of zamia and saying, look, we know Charles Chamberlain says that they're wind pollinated, but we're going to figure out what these weevils are doing. And so they did all of the necessary exclusion experiments and pollination behavior experiments. And they found that the plants are completely reliant on pollination by their pollinators. So if the pollinators are excluded, there is 0 seed set. If the pollinators are allowed in, it approaches 100% seed set. And this was in two different groups, two different species of plants. And in both cases, they identified the pollinator as two different species of this weevil, Rhopalotria. Also, one of this group of work was done at Fairchild Botanical Center, and there were closely related species of zamia nearby, and these weevils stayed faithful to their host species. So that group of researchers went further and worked out the life cycle of these pollinators that I'll just walk you through here. So the pollinators are housed on pollen cones. They aggregate in large numbers on pollen cones. This is the plant, the pollen cone. What they do is the females lay their eggs inside the microsporophyll, which is just a cone scale of the plant. And they burrow in with their rostrum and they lay their eggs in there. And they feed on the microsporophyll as adults as well. The larvae hollow out the microsporophyll. If one encounters the other, it cannibalizes it so that only one ends up inside each cone scale where it pupates in the stalk. It then emerges and starts the process all over again. At some point, they visit the ovulate cone, but these researchers didn't know why. They do not aggregate in large numbers and they do not feed on the pollen cone. An interesting thing to note is at this stage, at a late stage larva, they will go into diapause, which is just a resting state for the 10 months out of the year that the plant does not have reproductive cone. And so they just wait in this dried out cone until the next season when the plant is reproduced. And as people that are in Canada and myself in New York, you can imagine that Florida, where this work is done, is a very hospitable environment for living year round, especially as I look out my window at snow right now. Diapause is something that insects do in inhospitable conditions. And so inhospitability to these insects is to not having the presence of their host plant, even as I mentioned that closely related plants are nearby and could provide them housing. They stay faithful to their host plant. Here's just some of my pictures of the same thing. Here's a large Zamia furfuraceae. They're aggregating on cones at specific reproductive developmental stages. If you break a cone open, you see nibble marks and frass. This is the pollen cone. This is an ovulate cone that cracks open when it's receptive. Here's a weevil just covered in pollen. And this last picture is if you look at a pollen cone just head on and sort of lift apart the two cone scales, you can see a microsporophyll above with pollen sacs and standing on the bottom, standing underneath it. So that's where they live and exist in. This is an example of brood site pollination mutualism, or as some people call nursery pollination. So this is quite rare in nature, actually, and the examples that we always talk about are the fig and the fig wasp, or the yucca and the yucca moth. And in all cases, these are where both lineages are completely reliant on each other for life cycle completion. The insect raises its young, its brood, inside the reproductive tissue of the host plant, and the host plant is only pollinated by its mutualistic pollinator. So I wanted to know, I came into this as very simple questions. What are the mechanisms that underlie this relationship? What is that last piece of the puzzle? What causes them to move from the pollen cone to the ovulate cone and pollinate it? And so I asked very basically, what's the plant signal? What is the insect perception of signal? What is the insect's response to the signal? And how does the plant use the signal to enact reproduction? And of course, as I mentioned, there's visual signaling is not as important in these organisms because they don't have the large, colorful, showy flowers. So I expect this to be chemical communication. So to start off with what is the plant signal, I do something called headspace volatile collection. So what I have here is an inert plastic bag that is covering a pollen cone that is dehissing pollen. This filter here has absorbent material inside of it, and this is a vacuum that it's attached to. So all I'm doing is pulling air through this for 45 minutes where the chemical compounds will be attached into this mesh here, where I will then elute it with a compound and run it on something called gas chromatography mass spectrometry, which I'll just refer to as GCMS from here. All that GCMS does is separate all of the compounds that the plant makes into individual compounds so I can ask questions about those compounds. This is what that data looks like. So you have each peak here. This is the solvent peak at the front, but each peak is a compound that the plant makes. And it seems as though doing this, that the plants really seem to produce two main compounds, 1,3-octadiene and linalool, and that's it. So that's the plant signal, but I don't know what the insect perception to that signal is. So insects are different than humans. They don't just smell everything that's out there. They have to have odorant receptor proteins in their antennae that you could think of as a lock, and the odorant or the volatile is the key, and they must match in order for there to be a response. So I did something called electroantennograph detection to identify what the physiological capability of perceiving the plant signal is by these insects. So what I have here is this big thing in the bottom is the GCMS. So that's the thing that separates all my chemical compounds into individual compounds so I can ask questions about each of them. At some point it gets split, the sample gets split, half goes to the detector for me to identify it, and the other half comes down this tube where it's bathed over this antennal prep. So what I have here is a weevil whose little head I had just cut off. I'm sorry about that. These are two electrodes, and just like an electrical cord that you plug into the wall, I have the antennae attaching these two electrodes. When the volatile bathes down, when the volatile key bathes down and matches with the protein lock, then it closes this signal and creates an electrical charge. And because I have split this, I can match it to the chemical compound at the same time. And so this is what that data looks like. You can see here is the response and with the red arrow, the weevil, and on the top is the plant compounds. So this tells me that the weevil is capable of perceiving 1,3-octadiene, and it doesn't even know linalool exists, right? So that's the weevil response to that compound. We know in cycads that all cycads are thermogenic in their reproductive tissues, which means that they increase their own body temperature above ambient. And this is just a great image from the literature showing that, these cones here reaching about 28 degrees Celsius above ambient temperature. What cycads do, their thermogenic behavior is that at the time of reproduction, when the pollen cone is dehissing pollen or the ovulate cone is receptive, they will undergo a daily increase and then decrease in thermogenesis. And they do this every day for about 2 weeks when the cones are reproductive. We also know from some great plant physiology work of Irene Terry's that they undergo a thermogenic patterning that matches but is delayed a little bit from, a volatile production patterning that matches but is delayed a little bit from the thermogenic patterning. So they will undergo thermogenesis, and about 30 minutes or so later, they undergo an increase and then decrease in volatile production, not just of overall production, but they do change their compounds as well. And this is because thermogenesis does, of course, increase enzymatic activity and dissipation of volatile compounds, but also the thermogenic process creates the substrates that are necessary for the volatile production. So I hypothesized that there would be a behavioral response to differing concentrations of this 1,3-octadiene in the host plants. So to do the behavioral response, I do something called pit test. This is very simple. I have a different concentration of 1,3-octadiene in each of these cones and a control. I'm going to put 4 weevils in the top here of this Petri dish, put the lid on the Petri dish, and put the whole thing in the dark for 30 minutes. And just come back after 30 minutes and ask how many weevils have crawled down into the pit. When I do that, I see that the weevils are attracted. So 1,3-octadiene appears to be an attractant, but it is not linear. So you do not increase in concentration, which is on the x-axis here, and matchingly increase in number of weevils that have gone down into the pit on the y-axis here. So there seems to be 2 amounts where I see a big behavioral change. There's an attractant here at 10,000 nanograms of 1,3-octadiene, and it seems to be less attractive or even repellent at 650,000 nanograms. Now, this just tells me that their attraction level changes, but it does not tell me that this causes movement. And remember, what I'm interested in is there something that causes movement from pollen cone to ovulate cone? So for that, I built these little arenas, and I have mesh sides on all sides here to let volatiles dissipate. I have a side that has a my compound, or the side that has a control. And I just videotape these for 30 minutes, and I track their distance to each individual weevil. I track their distance to each one of the boxes, and then ask how that changes over time. And what I find is that they move towards, with the 10,000 nanograms, they move towards it, which is above this arrow, and they move away from it if it is this higher concentration. So there is an attractant amount, and there is sort of a repellent amount. So I want to ask then, how does the plant use that signal to enact reproduction? And my first question is, does pollinator attraction to the cone change throughout the day? We know that cycads are cell autonomous, which means that I can cut off a thermogenic plant cone, and its cone scales will continue to undergo thermogenesis and continue to undergo volatile patterning. So I've just set up these arenas with pollen and ovulate cone scales and a number of weevils, and I track their behavior over the entire course of the day. So these are the number of hours of the experiment, but you can really just sort of view this as hours of the day. We see right away that they are more attracted to the pollen cones than to the ovulate cones, and that there is a big change in attraction to the pollen cones over the course of the day. If I look at the volatile production of pollen and ovulate cones, I see that there is a characteristic patterning. So remember that this is percent composition of this compound. Green is 1,3-octadiene, orange is linalool. And remember, those are basically the 2 compounds that it makes. So it's going to equal 100%, the 2 of these together. So they're sort of the inverse of each other. But what's interesting here is that there is a dip in production sort of in the middle of the day, but after 8 p.m., it approaches 100% concentration of 1,3-octadiene in both pollen and ovulate cones. If I look at absolute amount that is produced, I have pollen cone here in blue and ovulate cone in red. And what you can see is that I've put on here the lines of the amounts that I tested, right? Or relative amounts to give you an idea of where they sit in here. But what you can see is that the pollen cones in general make much more 1,3-octadiene than the ovulate cones. And that here you can see, or here's my statistical value for the pollen cones also, both of them actually undergo a statistically significant increase in production of 1,3-octadiene at 8 p.m. This actually coincides with the amount of weevils exiting the cone. So for this, I had clear plastic boxes with mesh on one side with cones in it. And I videotaped it all day, and I just counted all day how many weevils had exited the cone, how many were outside of the cone in this box. And it would be 2, 2, 2, 2, 40, right? So at this point, they're exiting in mass from the cone at the same time that it's really producing much more 1,3-octadiene. So what we think is that there's a low level of 1,3-octadiene that the weevils just don't really care about. It's not really attractive to them. They don't quite notice it. And that's the amount that the ovulate cone exists at pretty much all day. Then there is an amount that is attractive, that is an attractant to the pollinators. And that's the amount that the pollen cone exists at pretty much all day until the end of the day, when the pollen cones increase in 1,3-octadiene into an amount that is now repellent to the weevils. But at the same time, the ovulate cones pop into the amount that is attractant. So a schematic of that would be this. You can see across the bottom is essentially a heat map, an increase in volatile emission. This would be over the course of the day. Oops, sharing is paused. Did you do that one, Carlos?
No.
Bring your shared window to the front. Let's see, sorry. There, is it back?
Good.
I don't know why that happened. But, and then if you think of it from the morning to the evening, you have a pollen cone here on the left that exists pretty much all day in this sort of purple mid-level concentration that is attractive. At the same time, the ovulate cone is not attractive. The weevils don't care about it. And so they are living on the pollen cone. But in the evening, as it heats up, it gets too stinky and the pollinator leaves that cone. But at that time, the ovulate cone now smells just right. And that's what enacts pollination. This is actually something called push-pull pollination, and the same mechanism exists in macrozamia for thrips pollinators. So we have this convergence in behavior from thrips and coleoptera, which are, of course, wildly diverged organisms. We also know that the thermogenic behavior is everywhere that it's been found. This is still questionable in Stangeria, which is a monotypic genus, so there's one individual here, or one species. But we also, everywhere that we have tested the volatile variation, we see that same patterning. So it seems as though this behavioral mechanism of the plant is consistent through evolutionary time, and we're having a convergence of behavior from the pollinators. So this chemical communication mechanism seems to be really important for this mutualism. It seems that this push-pull pollination mechanism is likely ancient and existed before the rise of flowering plants. And we know that the changes in volatile composition and over the course of the day describes the push-pull pollination. So this is a phylogeny from Michael Calhoun, and this is the species that we just talked about, Zamia furfuracea. So the obvious question is how are these traits evolving across this phylogeny? Is this indicative of coevolution in these lineages? So I asked that question for one other species pair. And as I mentioned, you need to have live weevils for this. So it isn't always entirely easy to do. I have a lot of species names going here. So this is Zamia integrifolia with its species Broclotrius lawsoni. The species names don't matter so much. And to not confuse everyone, I'm just going to call it the blue species pair and the red species pair. And I want to ask about trait evolution in these lineages. And of course, I'm asking about chemical communication. So my assumption is that chemical communication is driving these relationships. It clearly was in the species, in the blue species pair I already studied, but is it the same? Do I see the same behavior in the red species pair? And then I can ask, are these traits diverging across these closely related taxa? And what is the role of positive selection in trait divergence? So I do the exact same things with the red species pair. I identify the chemical composition of the plant. I identify the weevil's perception of it. The first thing I notice is that the weevils respond to and perceive this compound methyl salicylate. This plant does not make the compound we just talked about, 1,3-octadiene. I then do the same pit test and I see the same nonlinear pattern. As I look at the volatiles across the course of the day, I see the same pattern. This is displayed a little bit differently, but what you have is peak volatile expression hours and off-peak volatile expression. You see that in both cases, the pollen cone is making more than the ovulate cone. And you also see that the pollen cone increases in a huge amount during peak hours, and that the ovulate cone increases to what is presumably the attractive amount, which corresponds over here, of at the same time that the pollen cone is going into the sort of repellent amount. So it seems that this push-pull pollination behavior mutualism is the same across these species pairs. And as I mentioned, the plants make different volatile compounds. So we had 1,3-octadiene here in the blue species pair. We had methyl salicylate in the red species pair. So we're seeing this trait divergent in volatile production between the plants. And the question is there divergence in volatile perception in the organisms? So for that, I would take the blue weevil and give it a mix of blue volatile and red volatile and ask, can it perceive the red volatile? I take the red weevil and give it a mix of the blue volatile and the red volatile and ask, does it perceive the blue volatile? And when I do that, I see that volatile perception is diverging across taxa. So the red weevil is not capable of perceiving the blue plant's compound, the blue weevil is not capable of perceiving the red plant's compound. So the volatile perception is evolving to match the host plant volatile production. And in fact, some great work, some monographic work was done by Charlie O'Brien and Willie Tang in this plant or in this weevil lineage. And what they found was that there was not a lot of morphological variation in body morphology in these organisms, but there was a lot of morphological variation in antennal sensory pockets. And so we may be seeing a more rapid evolution in sort of this volatile perception, morphology, and phenotype in the weevils. But I wanted to ask, are the plants diverging across taxa? And so for this, I use living and museum collections to look at phenotypic diversity. So I worked with the Caribbean clade because we know that it's monophyletic. We also know that that's where my native Zamian tegrifolia, that's what's the red species pair. So I had done some work on that. These are all collection sites that the seeds were collected, wild collected, and then cultivated at Montgomery Botanical Center in Florida. So I'm able to collect volatiles from these plants in a common garden from their wild collected site. And then I used herbarium records to measure leaflet morphology across these populations as well. And so here's kind of a schematic of all this. So I've got this phylogeny that I built with transcriptomes that I'll talk about in just a moment. And here's exemplar leaflets. So you can see that there is some diversity that is interesting, right? Angustifolia is right on the beach. It gets a lot of wind damage. So we do see some variation in the leaflets here. And I measured length and width from a middle leaflet on each rachis. And then I use that ratio to take out any issues with size conflating the data. Then here, this is my schematic of volatiles. So this is that same phylogeny. Each row is a species. Each column is a volatile compound. And at this point, I'm treating these as traits. I'm not interested in identifying the actual compound. However, this red arrow is the compound methyl salicylate that we've worked out in the red species pair that I just spoke about. So each box, if it is clear, it means that this plant does not produce this compound at all. And increasing in intensity means that increases in the percent composition across all individuals in that species of each compound. So the darker the color, the more that species makes that compound. But my question was, similar to the weevils, do I see more diversity, more variation in volatile composition between species than I do leaflet composition? And so for that, I did something called an analysis of similarity. And I've got leaflet morphology on the top, cone volatile on the bottom with ranked dissimilarities on the side. But what we're really looking at here are these R values. So the R values in analysis of similarities, of dissimilarities, range from -1 to 1. Values below 0 mean that the organisms are more different within species than they are between species. And values closer to 1 mean that they are more different between species than they are within species. So what you're seeing is that we have a value above 1, but still fairly similar, fairly close to 1 in leaflet morphology. But we have a much higher R value in cone volatiles. So cone volatiles are more different between species than leaflet morphology is between species. And I wanted to ask if there's any role of positive selection in this. So I use the transcriptomes across all of these clades to do two things. To one, look for evidence of positive selection, and two, to bend the genes into 3 different functional categories based on their gene expression in that I won't talk about, but in their gene expression related to either reproductive development or volatile expression. And what I found was, this is the null hypothesis. This is all of the genes experiencing positive selection across all of the orthologous genes that I find across this clade. And your expectation would be that the positive selection would be sort of evenly distributed across these three functional bends: the volatile, the reproductive, and then all others. But what I find is that the others are a little bit depressed and that reproductive and volatile are increased. These values are not statistically significant using just a ratio test, which could very well be just because of very small sample sizes here. But it could also have, something to do with the setting up these bends. And I mean, there's a number of reasons that could be affected. But however, interestingly, within these these values of volatile production genes, I find two genes that have methyltransferase activity. That's their functionality. And methyltransferase, you can imagine being hugely important for production of methyl salicylate. And so this bin of genes gives me something more to look into for their function and sort of evolution across the whole lineage. So just to summarize that section, we know that now that chemical signaling seems to be a key mechanism of coevolution between cycads and their pollinators, because we know that distantly related weevils have evolved specialized responses to their host zamia, and that volatile compounds seem to be evolving more quickly than other phenotypic characters. And potentially there's positive selection acting on volatile production. And just for the last section, I remind you something I brought up briefly earlier, that cycads are highly toxic. There's two main compounds in cycads that we talk a lot about, BMAA and MAM. We know that they're mutagenic, neurotoxic, carcinogenic. We know that they're present in all cycad genera and all cycad tissues. And the toxic effects have been recorded in animals, insects, and very interestingly in plants. So I wondered, there's this, as I mentioned earlier, you can't just think of something as either a mutualist or a parasite, right? These pollinators are clearly mutualists. They're eating disposable tissue. They don't even eat the pollen. They just eat the pollen parenchyma tissue, and they provide reproductive services. However, they have an herbivorous behavior, and so they have a little bit of a parasitic behavior. So they must interact with these toxins in some way. So I wondered if there was anything I could glean from the evolution of what I'm going to term cycadivory, which is cycad feeding. We know a lot more about butterflies, so I started by looking at the parasites here. So these are parasitic herbivores of cycads. This is a phylogeny of all Lepidoptera. And I don't know if most people know this, but butterflies are nestled within moths. Moths are not a monophyletic clade on their own. So this is the butterfly clade here on top. But what we see is these at the top are all obligate cycad feeders. And we have some cases where there's been diversification. This Eumaeus lineage has diversified into at least 6 species. Xeranopsis lineage has diversified into 20-something species. And these are obligate leaf feeders. We see a lot of aposematic or warning coloration within them. We know that some of them do sequester toxins. These down here are more detritivores, but they have been found on cycads, these little moths. So I think that an interesting thing here is this idea that cycadivory evolved independently multiple times, and that there are many different potential outcomes. You can have diversification, aposematic coloring. And so I think that this is a great system. Cycadivory provides a great system for asking a lot of questions about, comparative questions about the evolution of species interactions. But in this instance, that I'm going to talk about today, what excited me about this was, all right, you can either independently evolve the ability multiple times to deal with these toxic compounds, or potentially you can just acquire a gut bacteria that does it for you. So I asked a very, perhaps even naive question, is, are there any gut bacteria that are shared amongst herbivores of cycad tissues? And if so, can we infer anything about the function? So for the paper, I sampled the cycad herbivores I could get my hand on. So I had some from Singapore, a leaf feeder from Singapore. I had 3 different populations in Florida, and then 4 different herbivores. I had a stem feeder. This is the stem in zamia. Most zamia is underground. So an underground stem feeder, 2 pollen cone feeders, and a leaf feeder. And I just asked, using their 16S sequencing, which is essentially just a barcode for bacteria species identification, how different are they? And I find what you would expect, that bacterial communities are generally distinct, meaning that each individual of a species, its gut bacterial community is more similar to another individual of its species than it is to a distantly related species, and that it doesn't have anything to do with feeding tissue. I looked at their core microbiome. The core microbiome is just, this is after filtering. These are all of the bacteria that are found in every single individual of a species. Other bacteria are present. There can be 100+ bacteria in a gut, but many of them are transient. So we consider a core microbiome as the bacteria that seem to be present in all individuals of the species. We find a pattern that we expect. We know from the literature that beetles have much larger microbiomes than butterflies. I don't know what's going on with this guy, but it's interesting. But we find 5 shared bacteria across all of these organisms. We can infer a little bit about their function. We were able to not to identify one at all. 2, we were able to identify to family, which we know is a family that includes extremophiles and a family that includes symbionts. One, we were able to identify to genus, which we can get some information about. We know that Pantoea can have functionalities of nitrogen fixation, toxin degradation, protein hydrolysis, which is important in our system because BMAA, one of the toxins, is a non-protein amino acid. We know that they can be vertically transmitted. The last one we were able to identify to species, when we know that it has some anti-cancer activities, that they can necrotic death of mammalian cancer cells, and we know that there's some nitrogen fixation capabilities. So these bacteria may be performing some benefit to feeding on this toxic tissue. But it was exciting enough to us that we went back out and collected as many cycad herbivores as we can. So this is the phylogeny of cycads on the top, geography on the bottom, and feeding tissue is colored by the plant here. So there's leaf, pollen, and pollen cone and stem in yellow. So you can see that we have Lepidoptera, we have Coleoptera, we have Thysanoptera, which is the thrips, and we have feeders of all of these different tissues. And we find these same 5 shared bacteria across all of these organisms. We want to know what the function of it is. So it's a project that I've started here with some collaborators, Paco Barona Gomez, who's a microbiologist and chemist, and Angelica Cibrian. They're both at Longhibio in Mexico. She's a population geneticist and cycad evolutionary biologist. So we have cultured these bacteria, the gut microbiomes, we have cultured in the presence of BMAA, an absence of BMAA actually, and watched how those communities of bacteria have changed over time. And we have sequenced these genomes now of these bacteria, and we've identified emergent biosynthetic gene clusters. So that's the way bacteria sort of create a function. They group their genes together, and that's a biosynthetic gene cluster, and that's sort of a function for them. And they have shared functionality that is present in all of them that is not present in their closely related species. And those are something called siderophores, which seem to be communication between bacteria. So there's a lot of interesting information that's going to come out of this about what the actual function of those bacteria are. So just to summarize that, there is a shared microbiome that has potential symbiotic functionality that may allow for herbivory of their toxic diet and therefore underlie the symbiosis. At the time, this was the first evidence of convergence of gut bacterial communities across a diverse set of herbivores that share a common host plant. So if we go back to our questions, we can now answer them in the framework of cycads. So if we ask what mechanisms keep us together, a symbiosis biologist, is going to tell you the same thing that your couples counselor, your therapist will tell you, is that it is all about communication. You have to make sure that your signal, what you're communicating, is clear, that your partner can perceive it. You have to see what their behavioral response to your signal is, and does it provide the outcome that you both want. But at the same time, you have to understand and recognize that there is some hidden diversity that we all share that really allows that partner to even have a seat at the table and be a part of that conversation and be a part of that relationship and that symbiosis. What type of symbiosis are we? I mean, I argue regularly that that it is a spectrum, not not black and white. And that if you're going to study species interactions in any way, you have to really think about what are the mutualistic traits of this organism and what are the parasitic traits. You can't ignore others, or you may be ignoring something that can give you a lot of insight into that relationship. What is the evolutionary trajectory? Well, it does appear in this system that the characteristics or the traits that have to do with chemical communication with the species interactions themselves are evolving quicker and maybe driving the diversification, or at least driving diversity within each of these lineages. And finally, has there been any evolutionary innovation? So clearly there are different ways to be a cycad herbivore and likely different ways to be a cycad pollinator, and that each of these different lineages allows us to ask some comparative questions to understand the different mechanisms that may be underlying these relationships and sort of the outcome of these relationships. And with that, I'll just end by thanking, and first and foremost, I thank Montgomery Botanical Center, which is an incredible place. I do almost all of my field work down there because they have the plants there and they're dedicated to research. I'm a postdoc currently at Cornell with 2 advisors, Chelsea Specht, a plant biologist, and Rob Raguso in the neurobiology department. So I'm continuing to study the plant-insect interactions and in cycads. The Cycad Society, if anyone is interested in joining, please just Google it, the Cycad Society. We send out newsletters, we do research grants. It's a great organization. And of course, I'm funded by the United States National Science Foundation, and there's a list of people that have helped me in a lot of this work. So with that, I can just take questions. I guess I'll go back to this one if anyone has any questions.
Well, thank you very much, Shayla. It will be an applause in the live talk. That was fascinating. Est-ce qu'il a des questions pour Shayla? Well, I will start.
About the specificity of the beetles in pollinating the Samia. Do you expect a specificity of this related to the neotropical species? Or
yes. So do you mean, do I expect there to still be specificity between each individual species of pollinator and its host plant?
Yep.
Yeah, so that's one of the things that I'm working on in my postdoc now. We have a number of years ago now, Willie Tang did some work where he used just 300 base pairs of mitochondrial sequencing to look at the phylogenetic relationship within these lineages. And even within the Caribbean clade that I spoke about, he identified 3 species. But I've already done a little bit more sequencing within those, and I find much more diversity than he had just in that. So I'm working now on phylogenies of all of these mutualists, actually. So there's the published phylogeny of Zamia. I've got a phylogeny of the Eumaeus herbivore, the Rhopalotria pollinator, and the Fraxinotha pollinator are all in the works. In fact, I just got the data back about 3 weeks ago. And now I just have to work through the bioinformatics pipeline. But I'm not sure. I think that, it could be comparable to fig and fig wasp, where we do see some pollinator sharing, but it may be a little more specific, but we won't know until we get some of that data. But fortunately, these are really difficult to get, but this collaborator of mine, Willie Tang, has spent his life collecting them and has, hundreds and hundreds of samples. And so we'll have to wait and see. But what I have seen is that on the Caribbean, even on the same island where you have populations of plants fairly close, I see diversification in the weevils of those populations. So it could very well be. But Panama, who knows, because there's so many plants.
okay.
Hi. Hi, can you hear me?
Yeah, please, Marta.
Hi. Well, thank you very much, Shayla, for the presentation. It was amazing, everything you did and the way you explained it. I was a question, maybe it's very basic, but I have no idea about the subject. I found super interesting that you said when you explained that the antennas, Juan Carlos, how do we say antennas?
Antenna.
Okay. Are different depending on the species, because the species, is adapted to the different compound. But my question was, why the plants have or create or generate different compounds? Do we know why, or?
Yeah, I think there's 2 great approaches to asking that, right? From the plant's perspective, if you want to keep your pollinator specific to you and you don't want to, you don't want to hybridize, right? You want to have specificity and keep your pollinator specific to you. You want to create a highly specialized channel of communication between them. And so you want your plant, you want to smell different than a closely related plant because you want your pollinator to stay specific to you, right?
Okay.
But from the other end of that, that means that in order for this to evolve, there has to be other cues that the pollinator uses to find a host plant, right? So there must be something else, which could be, and this is another bit of work that I'm doing now in my postdoc, it could be CO2. These are producing more CO2 as they're undergoing thermogenesis. It could be humidity because they're changing their humidity rapidly as they're increasing temperature. It could be visual, even though they're not flowers. So there are other cues that the pollinator uses to both remain faithful to its host plant, as well as to allow for diversification so that it doesn't, like go extinct if the plant goes extinct. And so I think understanding more about the pollinator physiology and behavior will answer some of those questions for us.
Thank you.
Mm-hmm.
So, and I have another question, Shayla. So, do you talk about octadiene methyl? I can't remember the name. The second compound. Do you expect that it's going to change depending on the clades of Samias across the phylogeny? Because
I do.
There are so many clades, and there are so many endemics that are so recent, and all that. But at the same time, Samia is not a very old genus. You know what I mean?
Right. Exactly. And so I think that one of the things that's most interesting to me is that this is the methyl salicylate, which is the one that's important for that species. And this is the clade of this, I mean, we don't have anything from Cuba here, right? Cuba is just difficult to get for us. So this is the monophyletic clade minus Cuba. And you see that like all of the others don't really make, they make very just trace small amounts of methyl salicylate. So clearly there's something happening even just within this individual clade. But what I'm doing now, and in fact, my project this year was supposed to be, I'm building these phylogenies of these mutualisms, and I want to look at how it plays out on the plant phylogeny. So I want to do a very similar thing, look at volatiles across the phylogeny as well as phenotypic traits. However, it's impossible to travel for the last year plus, but the collaborators at Montgomery Botanical Center have been collecting volatiles for me. And so they've been collecting across the Zamia phylogeny. And I won't have sampling like this where I have every individual within the clade that I sampled, but I will have sampling across the phylogeny to see how much it changes. And it's an interesting question to ask. Do you just have these sort of 3 or 4 compounds and you just kind of mix them up, right? Or do you have a lot of variation within it? So that's something that we're doing. And it gives us the ability also to ask questions like, if you increase in this box, do you decrease in this box, right? And so how are the volatiles themselves sort of compared and expressed relative to each other, which will give us a little bit more idea of the sort of evolution of volatiles within them. Do you know of any work that's been done in Panama of what the volatiles are?
I don't think so. There is nothing going on there about that.
Yeah.
So I have another question about the herbivores and the bacteria. So maybe I missed it. So where do they get the bacteria from? And also at what stage of life, they are these adults that you sequence, or they're, the young ones or that?
Yeah. So we don't know where they get the bacteria. We do know, and I actually forgot to mention this, is that these bacteria are shared amongst these herbivores and are not found in their close relatives. So
That's cool.
There are plenty of places that you could pick up bacteria. We would assume that there would be transient bacteria that would be considered transient. So like if you're feeding on a leaf, you know what these leaves look like in the woods. They're covered in moss and stuff, right? Like there's certainly plenty of bacteria all over them. But we would assume those to be a little more transient. So we don't know exactly where they come from. We can do some what we call oligotype analyses, where we ask within each species how different is the bacteria, right? And so when you have very similar, so like all of your sequences of species A are all very similar to each other, it's kind of a pattern of vertical transmission, right? Because you know, and we see that in 2 of the 5 bacteria. However, there's a lot of caveats when you're doing this work. You're working with, you know, 300 base pairs of something, you know, so what do you, how much can you trust that? So we don't know exactly where they're getting it. As far as life stages, I let's see, I circled what was not the life stage. So the adults of these butterflies do not feed on cycads. I mean, they're nectar feeders, right? So but I wanted to put it in because everybody wants to look at pictures of adult butterflies, not little worms, right? Little caterpillars. So I captured the tissue where they were feeding. So I've got larvae here. I had adults here because adults feed on the microsporophyll. For the Fraxinotha here, I had their larvae because they feed on the peduncle, actually. Yep. And then here we have, I had larvae because they're stem borers as larvae. An interesting thing though, is that if you think about that the possibility that they pick these bacteria up from the environment, you do also have this idea of what is your environment. And so for these guys, their environment, they don't move really much at all. They feed just on the peduncle. They don't even come out into the microsporophylls really. So they're just hollowing out this stem. So they have a very closed environment. And yet I can look at, let's see, remember they have a large microbiome. So I can look at all of the bacteria and ask how, the same question I asked before, the oligotype analysis, how different are these bacteria that are found, right? So there's 31 bacteria that are found in all of these. If it was just a pattern of the fact that they have a small environment and they pick these bacteria up from the environment, you would expect that most of those 31 would all show similar sequence, right? But instead, I find like lots of variation in the oligotyping analysis across these bacteria, except in those ones that I talked about that are within this 5 core microbiome. Some of them were really similar within each other. So
is there a possibility that they come from the plant? You know, there is a microbiome from cycads from China, I think, that they did different tissues, I guess. So maybe, I don't know if they might be coming from the plant because they are very close to it. You know, they're also like the eggs and the larvae are feeding into it.
Yeah, yeah, definitely. There could be something coming from the plant. I think that when you know, are all, I guess it would be an interesting question, right? If you were to look at the microbiome of the plants, is there some bacteria that is sort of shared amongst everybody? And how does that overlap with the bacteria that's shared here? And I don't know, you must know of Angelica and Paco's work down in Mexico. Yeah, and so she's done a lot looking at the bacteria and stuff in the colored roots. And so that's been a question that we've talked about, is trying to find anything that sort of overlaps. But it's like a side project of mine and a side project of their students, and there's coronavirus. And so things are just kind of.
Sure. Are there more questions? Marta?
If anyone, I'm sorry, Adriel, do you want to go? Okay.
Thank you very much for your presentation. I'm curious about how.
Can you speak louder?
Yes. Yes. I'm curious about to know because I read in the Montgomery Botanical Garden was an explosion of herbivory 1 year. I want to know if you have seen the same for the pollinators while you're working there, and if you think that maybe a higher temperature changing in the precipitation of the local area will affect the chemical compound signaling of these pollinators.
Yeah, we have not seen an increase in pollinators at Montgomery Botanical Center in numbers. And that I know the paper you're talking about, that was one with Melissa and I, that there's a ton, there's been an explosion of the herbivores down there at Montgomery Botanical Center. And those herbivores are also spreading on to other plants that they weren't historically on. So that's an interesting idea, because that's a great point. We do see in around the same time period, actually, we see a shift. So the pollinators are always historically noted as staying faithful to their host plant. However, in the last, I don't know, 8 years, maybe 7 or 6 years, we have seen small populations of pollinators on a few closely related plants. And so I'm looking at those to look at population genetics, but also look at their behavior to see what is it. And this goes back to my idea of what are the characteristics that make you stay faithful to your host plant, but what are also characteristics that allow you to sort of get out of the relationship. And so I've been looking at them from those perspectives, but that I really thank you for bringing that up, because there may be some comparison there of just sort of the environment that they've been in that is similar to what the butterflies are experiencing. And I can't believe I didn't put that together. Thank you for pointing that out. That's a good point. But we do know that with increasing CO2, you can have changes in chemical compositions. We know that with increasing in temperature, you can have changes in expression of it. So possibly there has been sort of a change in production of volatiles. I suppose that could be possible. But I think that there are other cues underlying it as well that could be related to the relationship.
And the last question, Mark, but we can stay with Shayla for more than 10 minutes. Well, thank you everybody else. But we can stay a bit longer. We need